students. So please join me in welcoming Cat Helms.
Thanks,
Thank you,
Kristen.
Cat.
Hello, thanks for coming today. As Kristen said, our topic is the yew, which has definitely been in the news recently, so we're going to dive right in with some headlines. So the yew has been in the news a lot, especially in the Idaho Mountain Express, our local paper, and these are some of the headlines about yew killing moose calves, elk, and I haven't seen anything about pets, but it's mainly the elk and the moose that are dying from yew poisoning. And so we all know that we shouldn't be having yew in our yards, and I'm going to explain why. All right, so the yew plant, the English yew, Taxus baccata, it's very distinctive leaves. These simple needle-like leaves are 1 to 2 to 5 centimeters long. They have lateral leaves with a central stem. They are planted usually for shelter, shade, and ornamental purposes. I had a huge one in front of my house when I lived in Sandpoint, Idaho. It was very old. I never had problems with it, but I wasn't there that long. So it is a slow-growing and long-lived conifer. It's got a single-seeded seed cone surrounded by a cup-shaped berry-like aril. This is a little bit unusual for the conifers. The Taxus genus includes 12 species found around the world. This is the Deafnog yew in Wales. This is the oldest living tree in Britain. It's 5,000 years old, and you can actually go visit it in the churchyard. So I thought that was kind of interesting. That tree's been around a very long time. It's huge. All right, so the yew plant has been known to be toxic for a very, very long time, since the beginning of the 2nd century BC. So some historical references to the yew include Julius Caesar documented Catavolcus, I have problems with that name, poisoning himself with yew juice. The Celts committed ritual suicide by drinking extracts from yew foliage and used the sap to poison the tips of their arrows during the Gaelic Wars. More recently, Agatha Christie's A Pocketful of Rye, which was published in 1953, describes murderous events at the Yew Tree Lodge. A little foreshadowing there. It happens that the tea of one of the main characters, I don't want to spoil it, is poisoned with yew, and the poisoner was able to hide the bitter taste of the yew because the tea was made with orange rinds, which also have a bitter flavor. So there's a good murder mystery there. But again, this is not an instructional podcast on how to murder people. Please don't do this. This is more for information and why you should be careful of the yew tree. All right, so all parts of the yew, with the exception of the aril, which is this berry that surrounds the seed right here, is toxic. So the toxicity does decrease, it does not decrease on drying, sorry. So trimmings are just as toxic as the actual living plant itself, and the highest toxin concentrations are actually during the winter. Okay. So the toxin itself is called Taxine B. It's a beautiful molecular structure right there. There are other taxines that are in the yew plant that are toxic, but this is the one that mainly does the damage, so that's the one we're going to talk about. What separates from other taxines is this nitrogen right here, which is hanging out pretty close to this benzene ring. So if you see that nitrogen, you know you're talking about Taxine B. So I think it's a beautiful structure, but I like chemistry things. All right, so Taxine B is the toxin. It is a cardiotoxic calcium and sodium channel antagonist. I'll talk about what that is and why that matters. It is the most potent toxic alkaloid known. 1 gram of leaves contains 5 milligrams of taxines, and the LD-min for humans is 3.0 milligrams per kilogram of body weight. So we've talked about the LD50 in previous lectures. That's the lethal dose that can kill 50% of a test population. This thing is very toxic, so we only really need to talk about the LD-min. The LD-min is the lowest amount of a substance that when introduced may cause death. So this is an extremely toxic alkaloid substance. Okay, I apologize for the sad picture. because this thing is extremely toxic. So Taxine B, these are the oral LD minimum values for certain animals that have a tendency to partake of the yew plant. So there's chickens, cows, dogs, goats, horse, sheep. The elk I could not find, but I'm assuming that the LD minimum is similar to the horse, because very little is necessary in order to cause cardiotoxic effects that lead to death in these large mammals. Okay, so what does exposure do? After you ingest this, and this is all for humans, I assume it's similar for other animals, but this is documented from human case studies. So after ingestion, 30 minutes to an hour, you will have dizziness, mydriasis, that's dilation of the pupils, nausea, vomiting, abdominal pain, tachycardia, which is an increased heart rate, arrhythmias, which are abnormal heart rates, and potentially convulsions. And again, I keep putting pictures of the yew up here so that you all can identify it. The arrows are very distinct. All right, after 1 to 5 hours post-ingestion, you end up getting bradycardia, which is a slowing of the heart rate. You get paralysis, respiratory failure, diastolic cardiac standstill, circulatory collapse, and eventually death. This is not a fun topic, but it's important. Okay, so why does this happen? So Taxis alkaloids are derived from steroidal lactones, and they directly affect the nerves and muscles of the heart. So to understand how cardiotoxins work, you need to understand how your heart works under normal circumstances. So we're going to do a deep dive into cardiology. Buckle up. In case you didn't know, that's your heart. So your heart is extremely important. It is the muscle that contracts in order to get blood to flow to your lungs and eventually to the rest of your body. Every cell in your body requires oxygen to work, and your heart is the pump that gets the blood to those locations. So the main parts of the heart that I want to talk about are divided into the atria and the ventricle. So the atria are up here on the top, the ventricles are down here on the bottom. The heart is actually an upside down organ. The apex is down here. It's a little bit funky, but now you know your geography, you know where you are in the heart. So the right and left side of the heart, the right atrium and the right ventricle pump blood into the lungs in order to pick up oxygenated blood. The left atria and the left ventricle pump oxygenated blood to the rest of the body in order to get all of the cells the oxygen that they need to survive. So we're going to follow the path of blood through the heart, because that's always a fun journey to take, I think. So we start in the extremities, and you have venules that are taking deoxygenated blood through the veins, and you're trying to get it back up to the heart. So once all of those veins sort of converge, they converge into the superior and inferior vena cava, which is this SVC right here, and that leads into the right atrium. So we're here now. It goes through the tricuspid valve into the right ventricle. From there, we go through the pulmonary valve into the pulmonary arteries that lead to the lungs. And there we pick up oxygenated blood. So when you take a big breath, you're oxygenating the blood. And once that happens, it returns to the heart via the pulmonary veins. And here we go into the left atrium, through the mitral valve, into the left ventricle. We go out through the aortic valve into the aorta, which is the largest blood vessel in your body. And once we are in the aorta, we go everywhere. So we go into all the different arteries and then into the capillary systems. We drop off oxygen and the whole system starts all over again. So that is the path of blood through your heart, and that's why your heart is extremely important. So the main architecture I want to leave you with is the atrium and the ventricle, left and right, because that's where we're going to move next to do a deep dive on electrophysiology of your heart. So this is the electrical conduction system of your heart. Your heart is beating in a very distinct rhythm, and that lub-dub that you hear when you listen to your heart. Sorry, I blocked the mic there, my bad. Happens to be close to my heart. So the electrical system is responsible for that lub-dub that you hear and the contraction of both the atria and the ventricles in order to get that blood where it needs to go. So there's some vocab that's important here. The word systole, you may have heard that when you're at your doctor's office and they're taking your blood pressure. It's systolic over diastolic pressure. So the systole is the actual contraction of the heart chamber, either the atria or the ventricle, and then the lower number in your blood pressure is called diastole, and that's actually the relaxation of the heart chamber. And the difference between systole and diastole, that's your blood pressure, and that kind of gives you a baseline of how your heart is performing. Okay, so let's talk about electrical conductivity of your heart. So it starts out with the SA node, which is the sinoatrial node, and that is, going for a little walk here, this guy right here. So this is where all of the electrical signals of your heart are initiated. And so what the SA node does is it initiates the electrical impulse. And you may have seen EKGs on TV or had an EKG done yourself, and you'll know this very distinct wave. This is called a PQRST wave. I don't know, that's just what they named it. And each little piece has a letter designation. So this is PQRST. And each one of those letters is indicating a particular moment during the electrical conduction of your heart. So it starts with the SA node, and you get depolarization in the atria, which causes the atria to contract. And so this is a P wave right here. Then you get a little bit of a flat line here. That doesn't mean that you're dead. It just means that there's a break in between the electrical signaling. And this is what happens when you get to the AV node, okay, which is right here. So SA node to the AV node. And once the AV node is activated, what's called bundle branches carry signals from the AV node down here to the apex of the heart. So the reason that you have these two separate nodes is because you don't want your atria and your ventricle to contract at the exact same time. You don't want those electrical signals to be crossing, or you're going to have a bad day. So anyway, so you have the SA talk to the AV, and then what happens is you get these things called Purkinje fibers in the bundle of His, and then they tell the ventricles to contract, okay? And once the ventricles contract, you get this PQRS wave, and that's the ventricles contracting. So that's how your heart is talking to the muscles and what are called cardiomyocytes, which literally means heart muscle cells. Any questions about PQRST waves? No? I'm overwhelming everyone with cardiology. So again, we go SA node to AV node to the bundle branches to the Purkinje fibers, and that's how you get your heart to contract. It's very exciting stuff. In case you were wondering what the lub and the dub noises are, if you ever listen to, if you ever listen to your heart, the lub is the closing of the AV valves at the start of ventricular systole. And if you remember, the AV valves are this guy and this guy, right, left. The dub is the beginning of ventricular diastole, and that's when the pulmonary and aortic semilunar valves close. They're a little bit harder to see on here, but this guy and this guy. So lub, dub, lub, dub. So if you ever want to listen to your heart, now you know what those heart sounds are. Okay, so now that we've looked at the structure of the heart and how the electrical signals are working their way through your heart to get it to beat, we're going to dive even deeper, into the cellular biology of how a cardiomyocyte actually works. The reason for this is it's on this molecular level that the Taxine B toxin actually does its devious work. So how your heart actually works is an action potential, which is an electrical signal, comes from your brain, and it tells your heart, you should beat now. And your heart says, okay, how do I do that? And so an adjacent cell will basically send over some signal, acetylcholine usually, and the voltage-gated calcium channel is open, which is what's happening over here. So this is a voltage-gated calcium channel. When you activate it, it allows calcium into the cell. Calcium then travels to what's called the sarcoplasmic reticulum, and some of it's stored there. A lot of it moves down into what's called myosin and, oh, my brain just stopped working. Actin, filaments, and this is a myosin. These are myosin cross bridges that are formed. So once calcium comes in, these little heads move, and you get this actin-myosin connection, and you get a contraction. So that's how you get muscle contraction in pretty much every one of your muscle cells. Cardiomyocytes are a little bit different. They're not quite smooth muscle. They're not quite skeletal, but that's another deep dive for another day. So once you get contraction, the calcium is released, and relaxation occurs when, so the calcium unbinds from troponin, that's it, and is used to pump back into the sarcoplasmic reticulum for storage, and it's also exchanged with sodium in order to maintain cell potential. So this is how a muscle contracts in your heart. It's a major deep dive for most people who are not biochemists, but I still think it's interesting. And in order to know how Taxine B works, you have to have a general idea of how all of this interacts. So why does this happen? Why do you get these weird cardiovascular effects? So Taxis alkaloids are sodium and calcium channel antagonists, and Taxine B is cardioselective. So this only goes after heart muscle. The rest of your muscles should be perfectly fine. So it binds to and inhibits the influx of calcium into the conductile and contractile myocardial cells, and when this happens, there's a change in the electrical activity of the heart. So when you bind to sodium and calcium channels in cardiac myocytes and you block them, there's an increase in cytoplasmic calcium concentrations, and this causes electrical and mechanical abnormalities in cardiac cells. So before we were talking about, oh, this is great, this is how it usually works, we love this. All right, this is what happens now. So once Taxine B comes in, it's going to inhibit the calcium influx here, and it's going to block calcium from leaving the cell, causing an increase in cytoplasmic calcium. And when you do that, you mess up how basic muscle cells function. And if one cell functions poorly, they all start to go. Because as you know, when a heart beats, it beats in unison. Ba-ba, ba-ba, right? So if you don't have cardiac myocytes talking to each other and sending each other proper cells or contracting at the right time, the whole system falls apart. So when you inhibit calcium influx, you slow AV conduction, that's atrial ventricular conduction, and you prolong what's called that QRS interval on an ECG. I might say EKG, they're interchangeable. It all stands for electrocardiogram. The K is the German cardio. The C is the English cardio, but they are the same thing. So when you increase cytoplasmic calcium, it leads to electrical and mechanical abnormalities because you're no longer able to get calcium out of the cell, and the electrical potential of the cell itself changes. So that's your deep dive on cardiomyocyte biochemistry. You're welcome. So let's talk about these EKGs, ECGs. Now that you know what it's supposed to look like with a normal PQRST wave, let's look at what happens on a Yew poisoning ECG. I know everyone's face just went, oh, and they should, oh. So in a normal ECG, this is normal sinus rhythm, and this is a 12-lead ECG. If you're an EMT or anyone in the medical field, you know how this is supposed to look and how you set these up. But I'm not going to go too far into how to set up and read an ECG. Just know that this is a normal one. Where's my thing? There it is. That's normal, okay? So normal sinus rhythm is about 82 beats per minute. There's these nice smooth and present P waves, these normal QT intervals. That one's looking pretty good. That is a yew poisoning ECG, so it is a mess. When I first looked at that, I was like, what am I even looking at? I can't even find P waves. The QRS complexes are just scattered. It's a disaster. So a yew poisoning ECG will present with wide and slurred QRS complexes with PR prolongation, and you also get what's called extended QT syndrome. I've actually had extended QT syndrome as the side effect of a medication, and it is not fun. You feel very tired, you feel very sluggish, you feel lightheaded, and it's not a good feeling at all. And this is not a happy ECG. We don't want that. So these come from case studies. So a lot of the data we have from yew poisoning and these ECGs come from suicide attempts, which is extremely tragic. And so this is what it looks like when you get these people into the emergency room. Okay, so we've talked about the electrical disruptions, so that's suppressed cardiac contractility, decreased SA node automaticity, because remember your SA node is the pacemaker of your heart. And so if your SA node doesn't work properly, your heart doesn't know when to beat. And then you get blocked AV node conduction. So we talked about how the AV node is the second in that electrochemical step, and the AV node is responsible for telling the ventricles to propagate that electrical signal to get that contraction. So these arrhythmias, an arrhythmia is just an abnormal heartbeat. So these arrhythmias may lead to decreased cardiac output, which means that your heart is not being very efficient when it pumps blood. So you're not actually getting a full force contraction, so that's decreased cardiac output. And these ventricular arrhythmias may lead to cardiac arrest and circulatory collapse. If you cannot pump blood to the rest of your body, your circulatory system eventually will collapse. There's just not enough volume of blood within the vessels themselves to support life. It's a real bummer, and it gets worse. So there's no specific antidote or evidence-based antiarrhythmic therapy. A lot of different things have been tried, like forcing atropine. There's just no scientific data supporting that actually working. In animals, the only sign of toxicity is unexpected death, and I came across that sentence many, many times, and it's a real bummer. The only thing that may be able to save you is rapid supportive care. And this includes supporting ABCs, which stands for airway, breathing, circulation, intensive care management, vasopressor support, that's in order to keep your blood volume high enough to keep your circulatory system from collapsing, temporary cardiac pacing, which is putting in some type of pacemaker to help your SA node function even though it's not actually functioning properly. And then ECMO, which is a bit of a last resort, and that stands for extracorporeal membrane oxygenation. So that's essentially having a heart and lung system outside of your body. That is a very intense process, but that's really the only way that you can support individuals who have been exposed to Taxine B poisoning. Eventually the toxin will clear, but it's a game. How long will it take to clear? It depends on the dose. Can you keep someone alive long enough until Taxine B is cleared from the system? That's the game you play. Okay, so that was fun and exciting. Okay, so how do you prevent exposure? Be able to identify the yew. Do not use yew branches or leaves as play items for dogs or for purchase for companion birds. There have been a lot of reports of birds who have had yew branches in their cages and they chew on the bark and it does not end well for them. And dogs can actually get the toxicity from throwing a branch from a yew tree because, like I said, every piece of that tree is toxic except for the actual berry of the aril. But I don't think that's a gamble you should make. There have been reports in the literature of children eating those berries, And as long as they don't ingest the seeds, usually they're okay, but there are no guarantees. So owners should dispose of yew trimmings by removing, burning, or burying them where animals cannot access them. Never eat any part of a plant you can't identify, and if you accidentally ingest the aril, do not consume the seed. If you think you've been exposed, contact emergency services immediately. In the literature, I also found that badgers can eat the seeds of the yew tree, which is very unusual because there's a high level of Taxine B in there. But it turns out there's some enzyme in their gut that prevents the actual digestion of the seed itself. So badgers are miraculous animals, and they can eat the seeds of the yew plant and not be affected. It's very strange. All right. So this brings us to our local legislature. So as of March 2nd, 2016, it is a crime to buy, sell, or own exotic yew in Blaine County. The Ohio Gulch Transfer Station will accept yew debris free of charge, but it must be separated from other yard waste. If you suspect you have a yew on your property, you can contact the Idaho Department of Fish and Game's Magic Valley Regional Office. That is their number. Kristen provided these pamphlets. So there is information in here about the yew and what you do if you suspect you may have one. So this is an excellent resource. But when in doubt, call these nice folks. They're more than happy to help you. All right, so is it all doom and gloom and bad news? No, it's not. So humans have found an interesting way to live with toxic plants, and we have found lots of interesting uses for them, so we can turn that frown upside down, I suppose. And one of them is called paclitaxel. So this is specifically found in the Pacific yew, and it's in the inner layer of the bark in very small amounts, and that's where it was discovered. And this compound is actually what's called a mitotic spindle poison. So when your cell undergoes division, which in cancer cells, it's uncontrolled cell growth, and so these cells are constantly dividing. In order to do that, you have to create what's called a mitotic spindle. And what that does is it organizes how the chromosomes are separated during cell division. So if you can poison that, you can prevent cells from dividing. And that's what this does. So this blocks mitosis trigger. If you block mitosis in a dividing cell, it triggers apoptosis, which is cellular suicide. So there are all of these checkpoints in place whenever you're trying to copy and multiply cells. And if something goes terribly wrong, the cell is programmed to kill itself. Not always, hence cancer. But this particular molecule blocks these mitotic spindles, so you trigger apoptosis and you go back to what's called the G0 of the cell cycle without cell division. So it reverts back to doing its normal little cell thing. So this makes it a really good chemotherapeutic agent. It has been shown in clinical trials to be effective against ovarian, cervical, lung, esophageal, pancreatic cancer, and also Kaposi sarcoma, which is a real problem in folks who have HIV infections. So there is some light at the end of this very bleak tunnel, and yeah, so paclitaxel is out there and it is FDA approved, so that's a bonus. All right, so if you think you've been exposed to yew poisoning or know someone or even suspect it at all, these are the numbers that you call. So that's the pet poison helpline. They also have the website, and then you can call the poison helpline right there. So I always like to end with those numbers. Again, this is all for information. This is not a how-to on how to murder people in your life. So here's a list of all of my references.